In this podcast episode, Dr. Zachary Knight, a professor of physiology at the University of California and an investigator with the Howard Hughes Medical Institute, discusses hunger, thirst, and thermoregulation. He explains the biological mechanisms behind craving food, consuming food, and feeling satiated. Dr. Knight also delves into the role of dopamine in food craving and consumption, as well as the function of GLP-1 and related drugs in weight management. Dr. Knight describes two systems in the brain that regulate food intake: a short-term system that controls meal size and a long-term system that tracks body fat levels. The brainstem, particularly the posterior part of the brain, is responsible for determining when a meal should end on a short-term timescale. Signals from the gut, such as gastric stretch hormones, play a role in signaling satiety. In contrast, the hypothalamus in the forebrain monitors overall energy reserves and body fat levels on a longer timescale. Leptin, a hormone produced by adipose tissue, serves as a key signal of body fat reserves. Leptin levels in the blood provide information to the brain about energy reserves, influencing hunger and energy expenditure. Leptin was initially hailed as a potential cure for obesity when it was discovered in 1994. However, clinical trials administering leptin to obese individuals did not result in significant weight loss. It was found that obese individuals often have high levels of leptin, leading to a state of leptin resistance. Subsequent reanalysis of the data showed that leptin was more effective in individuals with lower leptin levels. Leptin resistance poses a challenge in using leptin as a weight loss treatment, but there is potential for its use in specific subsets of individuals with lower leptin levels. Feeding behavior is divided into appetitive and consummatory phases. The hypothalamus, particularly agrp neurons, plays a crucial role in the appetitive phase, driving the desire to search for and consume food. These neurons are involved in the motivation to find and eat food when hungry. In contrast, brainstem circuits are more involved in the consummatory phase, the actual act of eating and swallowing food. Agrp neurons are not directly linked to motor circuits, but communicate with other forebrain circuits involved in motivation. Contrary to popular belief, dopamine does not solely drive pleasure-seeking behaviors. Dr. Knight explains that dopamine also plays a role in food craving and consumption. Dopamine signaling in the brain is involved in the anticipation and motivation to seek out food. Dopamine release is triggered by cues associated with food, such as the sight or smell of food, leading to increased food-seeking behavior. Understanding the role of dopamine in food-related behaviors can provide insights into overeating and obesity. GLP-1 is a hormone that regulates glucose metabolism and appetite. Dr. Knight discusses the development of GLP-1-based drugs, such as OIC and Mounjaro, for weight management. These drugs work by targeting GLP-1 receptors in the brain and gut, leading to reduced appetite and increased satiety. GLP-1-based drugs have shown efficacy in weight loss and are being used in the treatment of obesity and related conditions. The success of these drugs has paved the way for the development of novel treatments for obesity and diabetes. One finding discussed in the podcast was the discovery of the activity patterns of agrp neurons during feeding. Through experiments using fiber photometry, it was observed that these neurons predict the amount of food an animal will eat even before the first bite is taken. This predictive behavior suggests that AP neurons play a role in preparing the body for a meal and initiating the process of satiety before eating begins. A fascinating aspect of agrp neurons is their ability to integrate cognitive and sensory information to regulate feeding behavior. For example, when presented with appetizing food, the activity of agrp neurons can decrease if the animal anticipates more food to come. This highlights the role of these neurons in making real-time decisions based on environmental cues and prior experiences. The discussion also touches upon the relevance of agrp neurons in understanding eating disorders, such as anorexia nervosa. Individuals with anorexia demonstrate a heightened awareness of the caloric content of food, indicating a dysregulation in the neural circuits involved in feeding behavior. The hyper-focused attention on food calories and avoidance of eating could be linked to alterations in the activity of agrp neurons and related pathways. The podcast delves into the genetic underpinnings of body weight regulation and obesity. Studies have shown that body weight is highly heritable, with a significant portion of variation attributed to genetic factors. Mutations in genes related to agrp neurons and their signaling pathways have been associated with severe obesity in humans, highlighting the importance of these neural circuits in controlling body weight. The conversation then shifts to the interaction between genetics and the environment in determining body weight. While genetics set the baseline propensity for body weight, environmental factors can shift the distribution of body weights in a population. Changes in the food environment, such as the availability of highly processed and palatable foods, can unmask genetic predispositions towards obesity, leading to an increase in overall body weight in the population. Dr. Knight explains the concept of sensory-specific satiety, where repeated exposure to a certain flavor or taste can lead to a decrease in appetite for that specific food. He suggests that simplifying the diet and reducing food variety can help in controlling food intake. Additionally, he discusses how learning plays a role in food preferences and how the brain associates flavors with nutrient content. He mentions that highly processed foods with a wide range of ingredients may impair this learning process, leading to overconsumption. The discussion shifts to body weight homeostasis and the body's response to weight loss. Dr. Knight explains that when individuals lose weight, their energy expenditure decreases, leading to a lower metabolic rate. He mentions a study that compared the energy expenditure of reduced obese individuals to non-obese individuals, showing a significant difference. The concept of chronic deficits in energy expenditure for individuals who have lost weight is explored, raising questions about the long-term effects of weight loss on metabolism. Dr. Knight shares anecdotes of individuals who have successfully lost weight and maintained it by adopting a diet focused on minimally processed whole foods. He emphasizes the importance of behavioral regulation and lifestyle changes in achieving sustainable weight loss. He acknowledges that individual differences and factors such as alcohol consumption can also influence weight management success. Dr. Knight discusses the role of GLP-1 agonists in weight loss and their impact on appetite suppression. He explains that GLP-1 agonists were initially developed as drugs for diabetes due to their ability to increase insulin production in response to glucose intake. The discovery of GLP-1 in the intestine led to the development of drugs like exenatide and liraglutide, which have been approved for diabetes and weight loss. GLP-1 agonists work by suppressing appetite, primarily targeting neurons in the nucleus of the solitary tract and the area postrema in the brainstem. These regions receive direct input from the vagus nerve, which senses gut activity and influences hunger signals. By activating these neurons, GLP-1 agonists reduce appetite and promote weight loss. The guest explains the evolution of GLP-1 agonists, from short-acting formulations with a 2-minute half-life to long-acting versions like semaglutide with a 7-day half-life. These extended-release formulations have shown significant weight loss benefits in clinical trials, with some participants losing up to 16% of their body weight over a year. The podcast delves into the safety and efficacy of GLP-1 agonists, highlighting their unexpected health benefits beyond weight loss. Clinical trials have demonstrated improvements in cardiovascular outcomes, reduced inflammation, and even decreased alcohol consumption in individuals taking GLP-1 agonists. Dr. Knight emphasizes the importance of empirical evidence in assessing the impact of these drugs on overall health. Furthermore, the podcast discusses the potential for combination therapies to enhance the effects of GLP-1 agonists. Dual agonists like tirzepatide, which target both GLP-1 and GIP receptors, have shown superior weight loss outcomes compared to single agonists. The addition of glucagon in triple agonist formulations further increases energy expenditure, leading to even greater weight loss results. Dr. Knight highlights the effectiveness of bariatric surgery in weight loss, mentioning procedures such as stomach stapling and gut removal. He also discussed a promising compound from Amgen, referred to as AMG 133, which targets both GLP-1 and GIP receptors. Unlike other medications, AMG 133 is an antibody with a longer lifespan in the body, allowing for monthly injections and significant weight loss results. The discussion then shifts to the thirst mechanism and its connection to salt regulation. Dr. Knight emphasizes the tight link between the desire for water and salt, as both systems work to maintain the composition of the blood at the right concentration. Thirst is driven by specialized neurons that sense changes in blood osmolality, triggering the sensation of thirst when the osmolality increases. Dr. Knight's research on thirst neurons in mice revealed how these neurons track water intake and blood osmolality to regulate drinking behavior. Dr. Knight delves into the forebrain circuitry involved in thirst regulation, highlighting the role of the subfornical organ and the OVLT in sensing changes in salt concentration and hormones like angiotensin. He discusses how these neurons drive drinking behavior through negative reinforcement, making thirst a highly aversive sensation that animals seek to alleviate by consuming water. While Dr. Knight acknowledges the complexity of feeding behavior and the challenges of outsmarting the body's homeostatic mechanisms, he highlights the importance of making informed dietary choices based on scientific principles. He underscores the role of sensory cues, nutrient sensing, and gut-brain communication in shaping eating behavior, and emphasizes the need for a balanced approach to nutrition.